Welcome to University Lab Tech School. I'm Lori, the Lab Tech, and today I'm going to show you how to transfer broth to broth. This is what I do for our students. We have racks with pre-labeled the bacteria. We have them label the test tubes with a Sharpie. So another way you can do this is have labeling tape and label every single test tube with the correct bacteria. So there are 2 classes on Tuesday, so I'm setting this up for 24-hour incubation, and then I'm just going to put them in the refrigerator and we will pull them in the morning for the students. So we incubated these for 24 hours. Yesterday I showed you how to take the culture from the agar, and today you can see that they are grown very, very thick. This is E. coli. This is P. aeruginosa. This is the snotty one. This has a sort of a, see the snot in the middle? And it sort of has a green hue to it. When compared to something else, you can see the green. This is Bacillus cereus. Looks like E. coli, but it's different. Staph epidermis is usually clearer. You can see that this is way clearer than the E. coli. It's still got a lot of growth. And Serratia marcescens is that pink color. You can vaguely see the pink in a broth, but you can see it. See, comparing it. I guess it looks like a brownish color, but there's pink there. Okay, so it's really easy. What we're doing here is just taking a sterile transfer pipette, and we again will flame you can either flame the flask or the lid. I flame the lid, and I take up a little bit of this, or a full transfer pipette full, so a full mil, and I just will open these and quickly suspend them. These are all sterile broth, nutrient broth solutions. They're all in 2.5 mils of nutrient broth. And so what I'm gonna do is just squirt the right bacteria into each one of them and just keep doing it. And then I'll incubate these. It doesn't matter, you know, there's gonna be these are gonna be nice and thick and cloudy tomorrow, so it doesn't matter how little of a drop or how big of a drop I get, just that I remember what number I'm on. So that's why I lined them up like this. So I'm skipping one, and as I go, you're gonna see me move one back, and then I'm gonna drop the suspension in here, move it back, drop the suspension, move it back. Then this row, I'm gonna move it back here so it'll look nice, and I'll know which one I stopped on. Sometimes I feel like there's just not enough in like the last one, so I'll keep it and squirt a little bit more. You sterilize your bench in between the bacteria. So again, I like to put these trays out for when I'm done with like a whole, this is for the big basket, it goes in the big tray. When I'm done with my bacteria, I'm gonna move it aside. So I'm just gonna line it up for which bacteria I have first. I like to do the Staph epi first because, like I said, that can easily be contaminated. And then I'm gonna do P. aeruginosa, Staph epi, P. aeruginosa, B. cereus, S. marcescens, and E. coli. So before I start, let me just show you my setup here. So I have my bacteria, have my transfer pipette, my sterile transfer pipette. You can also use volumetric transfer pipettes, and you can also use like, you know, conical bottles, or like screw cap glass bottles. And here's my setup. Okay. Okay, I'm gonna get my flame going and not talking and zip through this. I will aspirate this up and down just to mix it, or you can mix the flask. You don't want to do it too vigorously or else it can kill the bacteria. There's two ways you could hold the test tube. I've seen people hold it like this, pour it in a few drops, and then so you lift the lid up while it's in the rack. It doesn't work with me. I like to lift it all the way up with one hand. Try not to touch the inside of the test tube. The outside of the test tube or the inside of the test tube. I try to just balance the pipette over the test tube. If I drop or touch this tip somewhere outside of the tube, then I'm going to throw it away. A little left over my tube. I'm just going to put it in my biohazard bag. And just give it a good one. Put it in the fridge. Okay, I'm going to move the flame. It's a little too close to me. It's too hot. Some goggles too. Cool. You're also going to want to change your gloves or disinfect your gloves too, just in case. But I can't work with the wet gloves, so I'm going to dry them. You can kind of look at them and see the broth already mixing in. Here's the clear one. See, you can definitely tell there's bacteria in this one and not in this one. If I touch something with this, I throw it away. Since I don't touch anything except for the inside of the test tube, then I'm reusing it, that's why. how you hold the test tube. I like to grab it with my pinky, lift up with my pointer. You need to maybe go higher with your pinky. You get a good feel for it. Drop a few. Put these in my bacterial refrigerator to use for the next class, and put all of these tubes into my incubator for 24 hours, and then take them out and put them in the bacterial refrigerator, ready for the class to pull on Tuesday morning. After a 24-hour of incubation, these are what our tubes look like. And I'm gonna go ahead and put them in the bacterial refrigerator. I like to shake these up. Look at all that growth. It's slightly pink. This is the one. And then this one has a snotty at the end and slightly green. E. coli is nice and uniform. Be serious. Sometimes it collects at the top there. When we mix it, you'll see. Staph epi, somewhat clear, but though this is a lot of growth for Staph epi, but I could still tell that it's clearer than the other one, so I didn't contaminate. So I put them in a tray, put them in my bacteria refrigerator for Tuesday AM lab, and I'll do the same for my Tuesday PM lab. Thanks for joining me at University Lab Tech School. I'm Lori the Lab Tech. Have a great day.